let's start. Hello, good evening everyone. I am Shakshi Bhardwaj, a PhD scholar under Professor Pradeep Kumar Maji, Department of Polymer and Process Engineering, Indian Institute of Technology, Roorkee. I am a course coordinator of Professor D. Maiti, Department of Chemistry, IIT Bombay, and welcome to the NPTEL problem solving session 4 for the topic Basics in Inorganic Chemistry. Here we are taking the assignment 4 in practice questions. This is the last practice solving session of this class. And let us start off with a brief introduction. As I already told you, my name is Shakshi Bhardwaj. I am in Department of Polymer and Process Engineering, Indian Institute of Technology, Roorkee. I am a PhD student under Professor Pradeep Kumar Maji, and my topic for the PhD is fabrication of cellulose-based aerogels for thermal management applications. Apart from it, I have already done two projects, one in Indian Institute of Technology, Roorkee, and another one in University of Delhi. Apart from it, I am also the Master of Science in Organic Chemistry from Maharishi Dayanand University, India, and Bachelor of Science in Chemistry Honours from Perodimil College, University of Delhi. So let's start with the session. From previous classes, we have learned a lot about CFSAs, John Teller, distortions, the basics of inorganic chemistry. Here in this session, we are taking some problems of lanthanides, actinides, term symbols, and then finally move with bioinorganic chemistry. So starting with the lanthanides and actinides. So this is the list of lanthanides starting with the atomic number 57 from 71. Yes, and this is a fifth period elements, mostly metals. And actinides, they are also 6 period elements. Actinides are metals, and most of them are, or all of them are, radioactive in nature. Whereas lanthanides generally occur in nature. Actinides are synthetic. Why we are taking this? Because we need to study about the lanthanides and actinides, and especially their electronic configuration, and we need to remember it for going to the next topic, which is term symbol. So start with the lanthanum cerium. Because of these are F block elements. I forgot this is main. They are F block elements. In this case, we are filling the first F. In the lanthanides, we fill 4F, and in the actinides, we fill 5F. In the case of from lanthanum, lanthanum is 4F0, and from cerium we start from 4F2 directly, and then 4F3, 4F4, 5, 6, and so on. Main is europium, in which europium and gadolinium, both are this consist of 4F7. Because of the stable electronic configuration here of the half filling of 4F, one extra electron will go into 5D, and here it is a 4F7 also. Similarly, with the case of yttrium and lutetium, yttrium has 4F14, and because of the stable electronic configuration of lutetium, 4F14 with 5 and one extra electron will go to 5D1. Actinides are filled according to the same criteria which we have filled with the lanthanides. So this is basic introduction of lanthanides and actinides. Now moving forward with the main topic, which is term symbols for an electronic configurations. So we actually define term symbols for any electronic configuration, and which is given by this formula, which is like 2s plus 1 L and J. This is in superscript, this is in subscript, and L is in between. S stands for spin quantum number, L stands for orbital angular momentum quantum number, and J stands for total angular momentum quantum number. To find out this electronic state, we first need to find out the electronic configuration of ion, and then in the case of F lanthanoids and actinoids, we need to calculate the number of F electrons in that particular case. For that, we need to memorize the lanthanoid series and their electronic configuration. And then finally, after calculating S, L, J, we can define this term symbol. Now question is, how can we calculate S, L and J differently? So there's a criteria to calculate all these factors, starting with the L. L is total orbital angular momentum. It is defined as sum of orbital angular momentum of each electron, which is given by this formula L plus 1 under the square root h upon 2 pi. Total L stands for total orbital angular momentum, a non-negative integer, and this is the basically scientific calculation for calculating L, which is given as the quantum numbers L1 and L2, and we can calculate the value by adding the L1 and L2 to subtracting the L1 minus L2. Or in the simple language, we can say that in the case of let us suppose S, we consider it as 0. If we talk about P, we can write it as 0, this is plus 1 and this is minus 1. In the case of D, we will write it as plus 2, plus 1, 0, minus 1 and minus 2. And finally, in the case of F, we'll write as plus 3, plus 2, plus 1, 0, minus 1, minus 2, minus 3. And this goes on for G, for H, and so on. So we will write them accordingly, and here's the terms. So for calculation of L, we first need to calculate the number of electrons in each of these orbitals. Like in this case, if we have 1 electron, so we'll consider it as plus 1, and the symbol for plus 1 is P. So here, so that term we need to calculate according to filling of electrons, and then calculate with the help of these numbers. With the help of these numbers we have written, and according to it, 0 stands for S, 1 for P, 2 for D, 3 for F, 4 and so on. So this is how we calculate L. Now S and J. So for S a spin quantum number, that means each electron has a different spin. Like for 1 electron we have a spin of 1 by 2. So that depends on the filling of electron. Like if we have unpaired electron, we will consider it as 1 by 2, and if we have, let us suppose, filled electron, this will consider it and this we consider as, or we will calculate all. So here it is, for each electron S will consider as 1 by 2, and therefore S is 1 or 0 for 2 electrons. And now we need to calculate the spin multiplicity, which is given by the formula 2s plus 1. And finally for the J, we calculate J with L plus S. We need to first find out the value of L, then S, and then by calculating we can calculate J. And yeah, J has the value of L plus S to L minus S. And in the case of half filled or more than half filled, we will consider this L plus S, and in the case of less than half filled, we will consider L minus S. So with these rules, we now move forward with the questions. This is the first example. It's a configuration like P2 configuration. So what will be the term symbol for P2 configuration? So here for P2 configuration, we first write down the filling of electrons like this, in which 2, 1 or 2, and by calculating this 1 plus 0, we got plus 1. So according to rule, 0 for S and 1 for P. So we got L, here is 1, which give us P. Now moving forward with the S, spin multiplicity, it has 2 electrons, so it has a spin multiplicity of 1 plus half, which is 1, and in the case of 2s plus 1, this will become 3. So now we got 2 symbols like 3, 1 is 3, 1 is P. Now here remains J. So what is J? So in the case of J, it is from L plus 1 to L minus 1. L we have 1, and S we have, in the case, 3. Oh, we have 1, and S we have also 1. We will consider S, this S, and this is L. So it is from 1 plus 1, which is 2, to 1 minus 1, which is 0, and in between there is only 1, 2, 1, 0. And because I already told you this is a case of less than half fill, we will consider it as L minus 1 in this case. Yes. So finally the term symbol will be 3, P and 0 in this case. Has anyone have a doubt? If anyone has a doubt, I can repeat this question again, this example again, sorry, and then if not, then we will move forward with the next question. Okay, quickly I'm repeating this example again. So here in the case of P2 configuration, we first need to write down the configuration according to the rule, which is this, and we will write the simples like plus 1 for this, 0 for this, and for the calculation of L, we will simply add these plus 1 and 0, which gave us the value of plus 1 and which correspond to P symbol. And for the calculation of S, because we have 2 electrons in this, so it is half of each. So by adding these, we will get 1. Spin multiplicity is 2s plus 1, which give us 3, a value of 3. So L is P, and the spin multiplicity is 3, so we got the 3P. Now the J, which we get by adding L plus L to L minus L plus S to L minus S. S here is 1, L here is also 1, so it's like 2 to 0. And because it's a case of less than half fill, we'll consider the value is L minus S, which is 0. The term symbol will be 3, P, 0. So with this, next question arrive. Like, what is the ground state term symbol for a D2 electronic configuration? For example, like Ti2 plus or V3 plus ion. So in the case of D2, what could be the ground state term symbol? Take 1 or 2 minutes to calculate the ground state term symbol, then we'll solve it here. Okay, till now I only get one answer, which is A. Okay, then we'll start solving the question then. Starting with the writing of this electronic configuration, first we need to make a diagram like this. And this stands for +2, +1, 0, -1, -2. And we will fill the electrons like this and like, because it has only D2 electron system, and this one correspond to this +2 and this one correspond to this plus. Hence L is equal to 2 + 1, which give us a value of 3, and 3 correspond to 0 for S, 1 for P, 2 for D, and 3 for F. So have we got the symbol F, and as from all the options there is only one F, which is A, so we can calculate the answer from here as well, but we will solve it completely for better understanding. So L is F. Now move forward with the value of S, because it has 2 electrons, so half for each, so it is half plus half, which give us a 1, and the spin multiplicity is 2s plus 1, which give us a value of 3. Now moving forward with the value of J, which is from L plus S to L minus S. So here from 3 plus 1 to 3 minus 1, we get a 4, then 3, then 2 here. So as it is also a case of less than half fill, we will consider the value of L minus S, which is 2. So the correct term symbol for this is 3, F, and 2, which is A here. This is the correct way to predict the sum symbols. Here is the explanation, simple, whatever we did today, filling of the electrons, then +2 plus 1 for, and then calculated the state, then going for the S, and then spin multiplicity, which is 3, the term symbol, state term is 3F, and for the value, which is L plus L, S to L minus S, from 3 plus 1 4 to 3 minus 1 2, and in the case of half filled, the lowest value of J, which will give the ground state term, so therefore the ground state term for the is 3F2. So hope this is clear. So moving forward, I have a similar question. So here, what is the ground state term symbol for the Gd3 plus ion? So I'm giving a hint here. That's why we study lanthanides, because it is a lanthanoid gadolinium we just now studied. also giving one more hint. This is has the electronic configuration XE 4F7 5T1 6S2. This will be the electronic configuration for this, and we will be written. So now we need to calculate the ground state term symbol of this. What could be the answer for this? Okay, someone has answered it as A. So let us start with the solution. So this is the electronic configuration of GD ion, but as we are talking about GD3 plus, that means these electrons will be removed in this case, and hence it is only left with 4F7. So we will only consider these 4F7 for this. And now this plus 3 plus 2 plus 1 0 minus 1 minus 2 and minus 3. So now we need to fill all the 7 electrons, which is like this. And now for the calculation of L, plus 3 plus 2 plus 1 plus 0 minus 1 minus 3, and here all will be cancelled out, and we will get the 0. 0 corresponds to the sum symbol S. Now for the S, spin multiplicity, it has 7 electron, and multiply by this half, you will get a 7 by 2 here in this case, and 2s plus 1 will be 2 7 by 2 plus 1, which gave us a value of 8. So we are having 8s. For the calculation of J, we got L plus LS to L minus S, and both is the case because we have a S of 0, so we will get only with the value 7 by 2. Hence the term symbol will be 8, S 7 by 2 in this case, which correspond to the option A. This is the answer. Correct answer is A. 8s 7 by 2 and has this gadolinium with the configuration. Then after removing 3 electrons we'll give the 4f and 7 electrons in 4f subshell. Each f contributed half, so 7f electronic contributed 7 by 2, which give the multiplicity of 8. Yes. And L has a value of 0 for S term and J from L minus S to L plus S. Because L we get 0, the J is equal to S, which is 7 by 2, and finally the term symbol will be 8S 7 by 2. Hope this is clear. And now we get to know that how we can calculate the ground state term symbol for any of the metal ion. Okay, so moving forward with the next question. Here, which of the following ions has the ground state term symbol of this? So this is a reverse question. Right now we are just calculating the ground states. Now we are given with the ground state and we need to calculate, or we need to find out which of the following ion has this. So for that we first need to know that all these are the lanthanoids. They are having what electronic configuration, so that we can cross calculate from them, which is having this 6H15 by 2 electronic configuration. So, okay, here's a hint. Giving all the 4f electrons of these ions, so later we can memorize them, but right now I'm giving you the hint as, and now we just need to calculate the ground state term symbol for each of the following and match which one of the following has this 6H15 by 2. I'm giving you 2 minutes to solve this question. Okay, I got an answer which says it's C. We'll see which would be the answer. Okay, let's start solving one by one. From EU, which is 3 plus, we have a 4f6 system, and for 4f6 system, we have filled like this: 1, 2, 3, 4, 5, and 6. Giving this plus 3, plus 2, plus 1, 0, minus 1, minus 2, minus 3. So by calculating plus 3, plus 2, plus 1, 6, and subtracting these 3, so we'll get 3, and which correspond to F, and it is a H, so this option can be neglected. Moving with dy, 3 plus, one, 2, 3, 4, 5, 6, 7. It has a 9 electron system, so we will fill this like that, and 9. So here's the 2 electrons in this state. So we can simply write 3 into 2, 6 for this, 2 into 2, 4 for this, plus 1, plus 0, minus 1, minus 2, minus 3, which give us a 11 minus 6 and 5. 5 corresponds to H. So this could be our answer. Let's see for the SM 3 plus. So we will fill 4, 5, 3, 4, and 5. 6 minus 1 give us a value of 5, which also corresponds to H. Okay, and in the case of TB, it has an 8 electron system. It is like this. So 6 plus 2 plus 1 minus 1 minus 2 minus 3. So it gave a system of 3, and which will, so this is not the option. So 2 of the options, this one and this one, got eliminated. Now we got with the 2, either P and either C. Now we move forward with the next thing, which is calculating S. So here in this case, what is the S? As we have these paired electrons, so we cannot count for this, so we only left with this, which will give us a system of 5 by 2, and the spin multiplicity for this case is 6, and here also S is 5 by 2. The spin multiplicity in this case also it is 6. So both of these are 6H. Now we only left the deciding factor, which is J, here also and here in this case as well. So J will decide which of these has this electronic configuration, and the value of J can be calculated by L plus S to L minus S. Both of these have L of 5 plus S of 5 by 2, and here also 5 minus 5 by 2. So this come a value of 15 by 2 to 5 by 2. Yes. So now Because dy has F9, it is a more than half fill, and SM has 4 than 5, it is a less than half fill. In the case of more than half fill, we will go with the value of L plus L. And in this case, we will get with the value of L plus minus S, which is 5 by 2. And this is 15 by 2. Correct answer we will get with this is dy. Okay, one of you in SM plus 3, L is 3, that is F. No, in that case L is not 3. In SM plus 3 case, if you calculate, we have 5 electron system. So in this case, plus 3, plus 2, plus 1, 0 and minus 1. So it is just minus 1 to minus 1, and we will got the value of 5, which give us a symbol of H. This is not 3, this is the case of H. In the S48 system and F6 system, we will get the value as F, not in the case of SM3. Okay, I hope this question is clear, that how can we calculate the ground state term system of all of this. So this is, if anyone have any doubt, I can repeat again. Okay, so if this is all clear, so now we are good with the ground state term symbols, and we can calculate the term symbols for any of the configuration by taking S, P, D, F and lanthanides, actinides and all these ions. So moving forward, the correct answer B, which we have already calculated. So moving forward with a new topic, which is microstate. This is a very easy topic. For a particular microstate is defined as one of huge number with different accessible arrangement of molecular motion energy. In other words, it is defined as each arrangement of energy of each molecule in whole system at a particular instant. How can we arrange? And it is also given with a formula, which is this: n x n minus x, where the n is twice the number of orbitals. Like for like we have p, so we can write p as it has 3 orbitals, so we can write it as 6. And if we have d, which have 5 orbitals, so we can write the value of n as 10. So it's a simple mathematical equation. So it's the 6 into 5 into 4 into 3 into 1, simple for 2 into 1, and 6 minus 2 to 4. 6 minus 2 is 4, 4 is to 3 is to 1. And by calculating, we can simply calculate the microstates. Now simple with this formula, we can have the question like calculate the microstate for a D3 electronic configuration. We have already solved for the P2. Now we need to calculate for a D3 electronic configuration. So what is the number of microstate, or what is the value of microstate? answer till yet. It's a simple formula based question. We'll write the formula again. This is n factorial on x factorial minus x factorial, whereas n is the number of orbital twice, x is the number of electrons, and we can calculate simply by this. Okay, someone has given us 20 and someone has given us 1140. We'll see what is the answer. So here the n for D is 10 because D has 5 orbitals, so 2 into 5, which gives us a value of 10, and we need to calculate for 3. So this is 10 factorial, 3 factorial, and 10 minus 3. So 10 into 9 into 8 into 7. I'm keeping it this because this is 3 into 2 into 1, and this is already 7. This will cancel out and we just need to calculate this. This is 5 and this is 3. So simple maths. By solving this, we'll get the answer is 120. In both of the case, it's not 20 either, or it's not 1140. It's simple 120 microstates. Okay, so as we don't have more questions for this, I will give you a question by myself. Calculate the microstates for, let us suppose, F4. What will be the microstates for F4? Calculate the microstates of F4 and based on previous. Answer yet. What will be the microstates for F4? With the formula n factorial x n minus x factorial. Okay, then let us calculate for this. is F. F has 7, just a 7 orbital system, and twice of this will give us 14. So the value of N is 14. X, we have 3 or 4 here. Okay, let's see. So it is like 14 factorial, 4 factorial, and 14 minus 4 factorial. So this is 14, 4 into 10, and we can write this as 14 into 13 into 12 into 11 and 10. We will keep this as 10 factorial, and below 4 into 3 into 2 into 1 into, already we have a 10 factorial here, so this 10 will cancel out this 10. This 4 into 3 gives us a value of 12. This 2 will cancel out this. So by solving, this is 7 into 13 into 11. So yeah, the answer is this 1 0 0 1. These are the microstates which this system will have. So I hope this concept of microstate and term symbol is clear, because now we are moving forward to bioinorganic chemistry. If anyone has any doubt related to these questions we have already solved, I can repeat either of them. Okay, if everything is clear, then we'll move forward to bioinorganic chemistry. So what is bioinorganic chemistry? So it's basically a biological inorganic chemistry. It studies inorganic elements as they utilized in biology. The main focus of these are on metal ions, where we are interested their interaction with biological ligands and their important chemical properties. So they can exhibit and impart to an organism, like in our body or in any of plants or animals' body. These properties include ligand binding, catalysis, regulation, sensing, defense, and structural support in our body. So our body consists of many metals, as we can say, like iron, sodium, potassium, copper. We have a lot of metals in our body as well, and they are functioning differently in our body. So the study of these metal ions with ligands or this consists of bioinorganic chemistry. This is an interdisciplinary field of chemicals which largely focus on the role of metal ions in living systems. And starting with, as talking about the bioinorganic chemistry, first thing comes into mind is porphyrin ring, or a porphyrin, which gives us to finally hemoglobin, which is an important factor in blood to carry oxygen in our body. So hence, starting with this system, so porphyrin ring is prepared by this 4-HCHO. It's a reacting these aldehyde with this molecule, and we need to get both these porphyrin ring, which we have 4, this molecule here, four pyros, and with this HCHO we'll get this system. And this position of 3, 2, 7, 8 12, 13, 17, 18, we'll consider this as beta. The position of 20, 5, 10 and 15, we consider it as meso. And the position of 1, 4, 6, 9, 11, 14, 16 and 19, we consider them as alpha. And similar to porphyrin ring, we have two more rings, chlorine rings, chlorine rings. The basic structure is this. Only difference in chlorine ring is that this double bond here get reduced and giving to the two saturated carbons. And in the case of chlorine ring, little bit change in the structure and in the saturation also. Here we are talking about these three rings mainly because they are very important. And yeah, because in chlorine ring there's a system known as, there's a phenomena known as chlorophyll, which happens in plant, and this consists of chlorine ring with, and the metal in this ring here is magnesium, and this helps plants to in photosynthesis. And another one is heme, which already we talked about, the ring porphyrin. If this molecule has iron in this will known as heme, and if this iron has more proteins attached to it, this will known as hemoglobin, which in our body in the blood carry oxygen. And last, in the case of chlorine ring, this is a chlorine ring. If it is having cobalt in its between, it is a vitamin, which is vitamin B12, also required for body. So these three rings with different three metal ions can act in our body differently. In heme, we'll discuss that in heme Fe in which state, either in +2 or either in +3 in the upcoming slides. So first to the question is, what is the central metal ion in the heme group? We already discussed heme group. This is not a typical question. All know the answer of this, which is, what is the central metal ion in the heme group? Simple. B. The answer is B, iron. Iron is the central metal ion in the heme group. These questions can come into exams directly. This bioinorganic is a very simple chemistry. We just need to memorize some of the basic facts, and on the basis of those facts we can simply answer these questions. So now, starting with the properties of porphyrin. It is a macrocyclic ligand. It is aromatic, planar pi conjugated double bonds follow Huckel rule, 4n+2, having 22 electrons, where n is 5. And we already studied that it is made of HCHO, and if it is replaced by PHCHO, then at meso position H is replaced by PH group. We already have talked about the meso position. We have these as a meso position. It is rigid due to the delocalization of pi electrons. It has completely delocalization of pi electrons without any unsaturation or saturation present in between them. Porphyrin molecule consists of unsubstituted tetrapyrrole connected by methylene diene CH bridges, and these positions are labeled as alpha, beta, gamma, delta, 5, 10, 15 positions, numbering of carbon only. The porphyrin ring can accept 2 hydrogen ions to form dications or donate 2 to form the dianion. It can also form cation, it can also form anion. In metalloporphyrin complex, the inner atoms, H atoms, are replaced by dipositive metal ion. So here in this case, metal-free porphyrin ligand has -2 charges. Porphyrin ring, the oxidation state of iron may be +2 or +3 in the heme group. This is what the respective question, that heme group is a porphyrin ring with iron atom at the center, and the oxidation state may be +2 and may be +3. and this is heme AB. We have different heme groups, like at the position of R1 and R2. If we have different attachments here, so this is known as, like in the case if heme A, R1 is replaced by this CH2 and R2 is having C1830OH. Similarly in heme B, R1 and R2 both have these group. Heme C, R1 R2 has this group, and chloroquine if R1 having this group, R2 having this group. Type A heme are found in cytochrome A. Type B hemes are like hemoglobin, myoglobin, peroxidase, cytochrome B. Type C are found in cytochrome C. Chloroquine is found in chlorocurine. All the biologist uses of heme group are important, but the most important is the binding of dioxygen molecules. The role of heme group is the binding of dioxygen molecule. Cytochrome also protein that contain a heme group, crucial for electron transport in cellular respiration and various other biochemical process. So simple question can be arise like which heme group, different hemes have different attachments, so they will give rise to different molecules. I hope this is clear. Moving forward, so this is the oxygen transport system in which heme group is present. The heme group is present, we know as hemoglobin. Heme group is absent, we know as hemocyanin, and both of them are used for oxygen transport. Oxygen storage, if heme group is present, then it is known as myoglobin, and if heme group is absent, it's known as hemurathrin. Directly question can come that which of the heme group can help in oxygen transport, and which of the following which do not have a heme group can help in oxygen transport. So basis on these, like if we have heme group, it is hemoglobin. If it doesn't have heme group, it is hemocyanin. And myoglobin, it is a Fe containing protein. Molecular weight is 1000 Dalton. Can coordinate O2 reversibly and control its concentration tissue. It is a protein chain containing 153 amino acids. Me contain iron in +3, high spin state of D6HS. So based on this information, let's start with the hemoglobin. We already discussed myoglobin. What is hemoglobin? Hemoglobin consists of a tetramer of myoglobin. Like if 4 of myoglobin units can attach together, then this will consist of hemoglobin. It has 4 sites, Fe sites, then can bind O2 cooperatively. The molecular weight is 64,500 Dalton. Hemoglobin is found in red blood cells called erythrocytes, and it is responsible for their characteristic color, which is maroon, maybe the color of blood. HP picks up O2. The role of hemoglobin is because it picks up oxygen from lungs, carries that in blood to the muscles, and where the oxygen is transferred to another heme containing protein MB, which stores it until oxygen is required. And it helps to decompose glucose to produce energy, CO2 and water. And then this CO2 is carried again to lungs. So this is the role of HP. It carries oxygen from lung to a different body, then from there it carries carbon dioxide again back to the lungs, and then we can respirate that carbon dioxide outside. So with the help of hemoglobin, our respiratory system as well as other systems will work. Each hemoglobin molecule is made of 4 subunits, each of which consists of a globin protein in the form of helix or spiral. The globin protein are 2 types, alpha and beta. Alpha globin protein has 141 amino acids, beta globin protein has 143 amino acids. This is the basic information about hemoglobin. So now the question, which of the following protein does not contain a heme group? Okay, this bioinorganic PDF I will upload these all presentations on the Google Drive. You can take that these presentations from there as well. I do not have the PDF of this because I collected all the data from different places and then just dump it down in this presentation. So you can take the presentation from the drive link. So which of the following protein does not contain a heme group? Already discussed that hemoglobin has a heme group. Myoglobin also has a heme group. Cytochrome C also has a heme group. Only the albumin which do not has a heme group. So answer is D. Albumin is a transport protein in blood but does not contain a heme group. Hemoglobin, myoglobin, cytochrome contains heme group involved in oxygen, either oxygen transport or electron transfer. So this is a simple question. Now what is the main function of heme group in hemoglobin? Electron transport, oxygen binding and transport, catalysis, or structural support. We already discussed about hemoglobin a lot. This is not a typical question. What is the role of heme group in hemoglobin? Okay, yes, the answer is B. Most of you have given correctly the answer. It is oxygen binding and transport. Hemoglobin, heme group bind to the oxygen molecule, facilitate their transport in the bloodstream. Each heme group can bind one oxygen molecule. Another question, what type of coordination geometry does Fe2+ adopt in the heme group when bound to the oxygen? When it is bound to the oxygen, what kind of coordination geometry does this Fe2+ ion adopt? So yes, the correct answer is B. These are the simple questions. We get octahedral, and in the case of octahedral, because why? Because already heme group has like this 4 nitrogen here, here. Okay, so I can direct it like this. Yes. So Fe has 4 nitrogen like this, this, 2 of which coordinates to it, and it has 2 remaining sides. One is this and one is this. From one side it will bind to the oxygen molecule, and from the other side it will bind to the different protein. So yeah, it has a geometry of octahedral. It binds to oxygen, coordinating with 4 nitrogen, a histidine residue, and an oxygen molecule. So this has a geometry of octahedral. So this is up to the hemes and the porphyrins and different. Now we will move forward to a different topic, which is iron sulfur proteins. So the iron sulfur proteins contain strongly bound functional iron atoms, but they are not porphyrin rings. They have iron and sulfur linkage. The iron atoms are bound by sulfur in this case, and these proteins all participate in electron transfer sequence. Their role is to electron transfer. FES protein play a crucial role in bioorganic chemistry, particularly in cellular respiration, electron transfer, and catalysis. So it has different kinds. So we are discussing some of them. Like first is dobridoxin. This is a simple but high functional iron sulfur protein. In this case, iron Fe is coordinated to 4 different sulfurs of cysteine, have going to give it a tetrahedral geometry. And the role of this is in electron transfer, particularly in anaerobic organism. Its simpler structure plays an importance in metabolic process, including hydrogen production, redox reaction in microbial systems. Okay, it's like Fe in which state? So mostly in this case Fe, I need to check, maybe it is a non +2 state, but I need to confirm this. This is not the point of concern here. In this case, the geometry or the linkage of iron to sulfur matters more. Similarly, here another one is ferredoxin, an important class of iron and sulfur proteins closely related to rubridoxins, but having a more complex structure. It is generally give rise to these structures in which irons are linked either to 3s, or here iron are linked to 4s. So this is a structure of Fe with 3s, 4s and 2s, and there are roles in their biological electron transfer. They are highly versatile, critical in various redox reactions, particular metabolism in prokaryotic and eukaryotic organisms. There are different type of these iron sulfur found in different organisms. Ferredoxins are essential iron sulfur protein which plays an important role in electron transfer, we already discussed, and they enable organism to conduct redox reaction in aerobic and anaerobic conditions. Their versatility and participation in various metabolic pathways make them significant in biological and biotechnological contexts. So these are rubridoxins and ferredoxins. Based on them, the question can arise: which of the following iron sulfur protein is involved in the process of nitrogen fixation? Okay, so it's a tricky question. It is not rubridoxin, ferredoxin, a different type of iron sulfur protein which is nitrogenase. Nitrogenase, as its name suggests, nitrogen fixation and nitrogenase. Nitrogenase catalyzes the conversion of atmospheric nitrogen into ammonia. And this is the nitrogenase. Nitrogenase contain the iron sulfur clusters like this as a part of its structure, and their clusters plays a crucial role in enzymatic ability, and they can transfer electrons for breaking the strong triple bond between N2, and the enzyme had two main protein. It is a 4Fe4S system, and here also molybdenum is present, contain the active site for nitrogen fixation, and a complex cluster that include iron, molybdenum and additional sulfur atoms. And the reaction mainly happens here is it can convert this into ammonia, and that is a process of nitrogen fixation. This is directly a new topic, and the question can come either in any form like this question, in which the following iron sulfur protein in the process of nitrogen fixation, and question could become like, in nitrogenase what are the metals present? Apart from iron, there are also molybdenum present in this case. Fe and Mo. There are two metals in the case of nitrogenase. I hope this iron sulfur proteins, and they have like question could become like what is the function of iron sulfur proteins. So we can tell that iron sulfur proteins has a function of electron transfer. So here is another topic, which is metalloenzyme. They are simple topics. We just need to memorize them for get the simple questions. So metalloenzymes are enzymes which are large protein molecules, and they have at least one reactant substrate catalyze an important biochemical reaction. So these compounds are extremely efficient at catalysis, and they can increase the reaction time to 106 or more compared to uncatalyzed rates. So there are also usually highly specific catalyzing only one or a few reactions. So now we will discuss one by one, that because question can come like this, which of the following is iron, copper or something like this. So first of all, iron containing metalloenzymes. So there are some like cytochromes, ribonucleoreductase and peroxidases. They contain iron in them. Cytochromes, these enzymes are involved in electron transport, in cellular respiration and photosynthesis. They contain iron in a heme group, which can undergo reversible oxidation and reaction from Fe2+ to Fe3+ to facilitate the electron transfer. The ribonucleotide reductase, this enzyme which contains iron sulfur cluster, is involved in the conversion of ribonucleotide to deoxyribonucleotide, an essential step for DNA replication and repair. In the case of peroxidase, these enzymes contain heme groups and help degrading of hydrogen peroxide by transferring electrons to it. So there are some iron containing metalloenzymes. Yes, now here come the copper containing metalloenzyme. Copper containing metalloenzyme generally or mainly the two, cytochrome C oxidase and superoxide dismutase. Cytochrome C oxidase is a crucial enzyme in mitochondrial electron transport chain containing copper and heme cofactors. It transfers oxygen to form water, essential for aerobic respiration. And the superoxide dismutase, copper zinc, it contains either copper, zinc, superoxide dismutase or manganese superoxide, and it can help protect cells from oxidative damage by converting superoxidase. Just a second, we'll check. Superoxide radical into hydrogen peroxide, H2O2 and oxygen. This superoxide into hydrogen peroxide and oxygen. The role of this superoxide dismutase. Moving forward, some zinc containing. Zinc containing is carbonic anhydride, which contains zinc at its active site catalyze the reversible conversion of carbon dioxide to bicarbonate and protons. Vital role in maintaining pH and gas exchange. Alcohol dehydrogenase, in which the inconversion of alcohol to aldehydes or ketones will take place. These are having zinc in them. Some manganese containing are manganese superoxide dismutase. We already have did this in superoxide dismutase. It is found in mitochondria, and it can help in protecting the cell from oxidative stress by converting superoxide radicals into hydrogen peroxide and oxygen. And photosystem second contain a manganese cluster for the light reactions of photosynthesis when it catalyzes the splitting of water molecule to release oxygen. Molybdenum, we already studied nitrogenase, which help to convert nitrogen into ammonia. It is molybdenum in it. And sulfite contain molybdenum and reduce sulfite to sulfite, and it's playing a key role in sulfur metabolism. So here this is the basic information about the metalloenzyme. I hope these are clear. Question can arrive either on the function of these metalloenzyme or either simple which of these metalloenzyme contain which metals, or like this. So this is the question: which of the following metals is most commonly found at the active site of carbonic anhydride? We already read about the carbonic anhydride. So which of the following metal is most commonly found at the active site of carbonic anhydride? got one answer, which is B. And yes, answer B, zinc is correct. Zinc is found at active site of carbonic anhydrase. Another question is, what is the primary function of superoxide dismutase? We already read it, the primary function of superoxide dismutase. What does SOD do? They are simple questions based on the concept of knowledge. Yes, answer C is correct. The superoxide dismutase converts superoxide radicals into oxygen and hydrogen peroxide. Last question on this topic: what is the role of zinc in the enzyme alcohol dehydrogenase? What is the role of zinc in alcohol dehydrogenase? We just read it about the alcohol dehydrogenase. Its simple answer is A. It stabilizes and structure and involved in the oxidation of alcohol to aldehyde or ketones. Answer is A. For this question, some simple questions like this can come from the metalloenzymes. Moving forward, like which condition results from excess copper leading to liver damage and other symptoms? This is a different topic than we have studied. This is based on disease. Like if we have some excess of some metals or deficiency of some metal, like deficiency of iron can cause anemia. We know that very well. Similarly, what test conditions result from excess copper leading to liver damage and other symptoms? Okay, the answer is A. Wilson disease. Wilson disease is a genetic order in which the body cannot properly eliminate extra copper, and as a result it can accumulate in various tissues, especially in liver, brain and cornea, which can cause liver damage to our body. So this is a Wilson disease. This is a very famous disease. This question can come several times, like what could be the disease from excess of copper. Okay, which of the following is the most abundant element in the human body? From these elements, which of the following we have most? Okay, we got the answer is carbon, but no, here the answer is oxygen. We have oxygen, the most abundant element in human body, because it's mainly found in water, can be responsible for respiration as well as involved in the energy production. So this is the most abundant element in our body, oxygen. This is not carbon. And yeah, similarly like this, we have come to the end of our sessions. Okay, I write it wrong. All the best for week 4, assignment 4. So we have covered most of the basics in inorganic chemistry from last 4 sessions. I hope these sessions will be informative and has informative for you. I wish you all the best for upcoming exam and for the assignment 4. Thank you so much all.